Great. With that, I think we'll switch over to our today. So it's my pleasure to introduce Jaz Millar. They're doing research at the University of Cardiff, but also based with the Natural History Museum in London. Also a final year PhD student, and I'm very excited to hear more about this research about microbes and the polar regions. So please do take it away.
Great. Thank you for that introduction. I'm just going to share my screen now. Yeah, thank you for coming. My name is Jaz Millar, and yeah, I'm working with Cardiff University and the Natural History Museum. Now I have to just change my own view. Yeah, here we go. So I can start clicking again. Great. I grew up in Cornwall. This is me holding a velvet crab and Plymouth when I was doing my work experience with the Marine Biological Association. And that's kind of how I got into the kind of science that I did. My first molecular microbiology project was when I was doing my schoolwork experience at Plymouth Marine Laboratory, and I'm still doing more or less the same science as I was doing there. But now I am working between London and Cardiff. I'm in Cardiff right now, and I'll talk a little more about why that is as we go along. So to understand my research project, it's a little bit of a history lesson. Often how we put deep history into context is by imagining it as a 12-hour clock. So if we're starting at 0 basically, then you have the formation of the Earth, and then quite quickly, just after 1, you have the first life, which would have been something like the bacteria we have now, in that it would have been quite simple and single-celled. Then before 3, we have the first photosynthesis, so organisms have started to use energy from the sun. And then by 5, we have more complex cells, eukaryotes, which is more similar to our cells and the cells of fungi, more complex organisms like that, and cells beginning to work together a bit more. But there's still no multicellular life as we know it. Then we have a period that scientists like to refer to as the boring billion, because you can notice that there's a bit of a gap on my clock here. I think that's quite a rude description because a lot of things happen with algae evolution. But of course, what people are really interested in is the Cambrian explosion and what happens from that point on when we have the evolution of animals, plants, and eventually at just before 12 o'clock, human beings. So the question is, what is it that took us out of this boring billion? What was happening during this time? How did we go from millions and millions of years of single-celled life into a world that looks like it does today? This period is between 720 to 635 million years ago. In the Tonian that comes before, we're pretty confident that things were still single-celled. All of the fossils that we have show that, and we don't have any evidence to suggest otherwise yet. But afterwards, in the Ediacaran, then we have soft animal fossils appearing in the record. So it must be this period in between that was the significant one on transitioning from simple single-celled life to complex multicellular animals. And this period is called the Cryogenian. Some of you who are more up on your classical languages may have already figured out something about the Cryogenian. It's also known as Snowball Earth. It's the main period in Earth's history where we can be pretty confident that ice came all the way down to the equator. And as I was saying, this is over about 100 million years. It probably happened in 2 large periods. So not only did life have to survive through this Snowball Earth period, it had to have been strong and diverse enough to stage the conditions for the first ever animals, one of the most important times in evolutionary history. So one of the main research questions of my current project is, how did life survive on a Snowball Earth? I've broken this down into some questions that I have, research questions that I've gone through for my project. The first one is, what key habitats supported life? We can look to understand how life survived in ice in the past to how ice survives in the present day. Here are some example habitats that we commonly look at. So on the left, we have ice algae on the surface of the snow and underneath the ice. But as you can see, it usually comes in blooms. That means there's a lot of it in density, but it's just one type of algae or a couple. You know, it's not the kind of biodiversity that's going to be staging this big evolutionary change. Same with the subglacial streams. You don't really get much photosynthetic light-loving organisms there because they're under the glacier for most of the time. Usually wherever there's water, there is life. So there are things living underneath glaciers, believe it or not. But again, not hugely diverse because they are trapped under a glacier in quite fast moving water sometimes for a lot of the time. Hydrothermal vents are became quite popular as an ecosystem that can survive anywhere, even at the poles, even at the depths of the ocean. And this is true, they are quite important at the poles. However, it's now been shown that they are fed by photosynthetic organisms that live above and fall down below to feed it. So actually, if the oceans were frozen over, then hydrothermal vents would not be the diverse ecosystems that they are today. And then we come to the last one I have, cryoconite holes. I'm going to just take these out and focus them, partly because you might not have heard of them before, they're a bit more obscure, and partly because they are a lot of what my research concerns. Cryoconite holes are depressions, little puddles on the surface of the glacier that are formed when matter blown in by the wind or deposited by stream, sediment, cells, biological matter, kind of muddy, sedimenty dirt lands on the surface of the ice. Then the sun warms that darker color and it melts the ice below it, creating these little puddles. Because of the darker color, it absorbs the solar radiation and melts down into the ice. So you have these little pockets, these tiny little pond habitats. Unlike the other habitats I mentioned, because these are a glacial surface ecosystem, they can support photosynthesis. They could still survive if the ocean became inhospitable, if the oxygen levels became very high or very low in the ocean. Things on the surface would be a lot less directly impacted. And they survive freezing. And there is proof of this in the field and in the lab. If you freeze these organisms, even for years, and then warm them back up, a lot of them will survive. So they're a really interesting candidate for a snowball Earth habitat. They're maybe one way, one refuge, one oasis for life from back then. So then the natural second question is, what organisms live there? And are maybe does that give us some clues to the kind of organisms that were living on Snowball Earth? So this is a question that I've been trying to answer for the last few years. And I've just at the moment, I'm coming to the end of writing my paper about it. Hopefully that will be going off to a journal very soon. I tried to get as wide a breadth of cryoconite as I could from across the poles. So I have 3 Arctic sample sites, Sweden, Svalbard, and Greenland, and a whole range of Antarctic sites. This bigger map you can see is a zoomed in view of the McMurdo dry valleys, where a lot of cryoconite is collected. And all of those points are separate glaciers, so this covers quite a large area. But I was also lucky enough to be given some samples from a more obscure site from some friends in Leeds. They went all the way into the Østeinen Scoop, which is much further inland. So we were really lucky to get some quite different cryoconite from there as well. So these are the samples I have to play with. I was actually lucky enough to go to Svalbard myself in my first year. So I have been doing some studies on my own samples, which is really lovely. The rest are from my supervisors and collaborators. Obviously, you do have to be a little careful when working in Svalbard because you never know when a polar bear will be around the corner. This was taken by my friend Roslyn when she was stood next to me, but she had a better camera than I did, so I always use her photos. But yeah, very lovely, very dangerous, which did actually impair some of our ability to go out But I was really there for polar fieldwork training, so I was really lucky that my supervisor happened to be in Svalbard and took me out to get some samples. It was a very nice trip. So back to my question, what's living there? How do we go from this sediment I've collected with my very scientific turkey baster that you can see there to actually understanding what organisms are living there? Back in the day, we probably would have done microscopy. And when I was talking to some school students earlier, I did show them some beautiful microbe pictures of how you do microscopy, but hopefully you'll have other speakers who can show you that, who are microscopy geniuses. But there are a couple of limitations to microscopy. One of them is that you have to be quite experienced to do it. It's not an easy job. It requires a lot of time, a lot of patience, and a lot of expertise, because a lot of these cells are incredibly similar. And some differently shaped cells can be the same species. Cells that look the same can be different species. So nowadays we use DNA methods to tell the differences. We extract the DNA. I extract the DNA, this is my job, from the sediments that I find from the cryoconite. And then we do what's called gene amplification, where we're basically making loads and loads of copies of a particular gene. The reason we're looking at one particular gene is that we pick one to be a kind of marker. We picked something that doesn't change too much, so it isn't lost between any species, something that all species have, but that there's just enough difference to be able to tell different species apart. So we pick this highly conserved gene and copy it up loads of times to the level where our equipment can detect it. We then clean it up. So again, we have a nice clean sample of just this one gene. And I did that twice. I got a gene to identify the prokaryotes, which is the bacteria and the archaea, the more simple celled organisms. And then another gene which detects animals, fungi, the more complex celled organisms. Then we can look up a database and line them all up and find out what species they are, if it's a previously recorded species, or at least what they're closely related to, if it's something difficult to sequence or something that is a new species. And we found so many different organisms in every single cryoconite hole from every single location. We found a huge diversity of microorganisms. We found something like 35 different phyla, and phyla is the most general group that you can give to any biological organism. So this is like, when you get down to species level, we're talking about hundreds. This included a broad range of stuff. We've got the algae over on the left here, iguanoflagellates, which kind of behave like incredibly simple animals. Bacteria with different properties, some that like oxygen, some that use oxygen, some that photosynthesize, and a load of different eukaryotes, including protists like amoeba, and microanimals like rotifers and tardigrades, who have kind of come into their own fame as being a well-known microanimal that can survive extreme environments. People have even taken these little mini animals to space and subjected them to all kind of torture, and they can survive a lot of things. So huge range of organisms that we found here. We can then line this back up to the organisms that we know evolved and were around by the time of the Cryogenium. Because of fossils and because of DNA tracing back lineages, we know that by the Cryogenian, as I said, we already had algae, we already had a load of bacteria, we already had amoeba, we already had iguanoflagellates. We know this for pretty sure. And then we know at some time near afterwards, then we think that we have the first animals, like I said. So it's really useful to know that cryoconite can support all these same things. All of the organisms that were alive during Snowball Earth can be accommodated by cryoconite. That's really good to know. That's really good evidence that they are a potential habitat that could have been around then, because it's housing the exact same types of organisms. Not all cryoconites are the same. We didn't find all the same organisms in the same ratios in every cryoconite hole. We just found a diversity in function and species. Some cryoconite holes in colder areas, like the McMurdo Dry Valleys in Antarctica, have thick ice lids that can last months. and they have thick sandy sediment at the bottom, they're much more isolated, and that will influence what organisms do well there. In the Arctic, we found sticky granules held together by cyanobacteria rather than sandy silt. The holes were open and connected to each other, both at the surface and sometimes underneath the surface, and that will influence what does well there. We found a striking difference between the Arctic and the Antarctic. So clearly the environment and the location influences the community, which is important when you're thinking about climate change now and climate change in Snowball Earth. But when you have a change to the environment, you're going to have a change to the community if different environments have different communities. So that's a bit about what's living in these areas. But of course, Snowball Earth is not the modern Antarctic. However similar it might look in photos like this, there are differences. My next question is a very classic science question. Can we replicate the habitat in the lab? Because we acknowledge there are these differences between the question that we have the simulation of Snowball Earth and a real Antarctic summer, can we get a bit closer? So the reason I'm saying Antarctic, by the way, rather than polar, is because the Antarctic habitats happen to have quite a lot of the organisms that I was particularly interested in. So I have used those in the following experiments, but I still look at both poles. Okay, so can we replicate this habitat in the lab? I've tried to replicate Snowball Earth conditions even more accurately than what we find in the field. So the first difference that I thought about between the modern poles and between Snowball Earth is that I'm imagining if the whole Earth was frozen over, most life would probably live at the equator where it's warmest. The poles would be much colder than they are now. So that means rather than a summer of light and a winter of darkness, like you normally have at the poles, there would be 12 hours of light and 12 hours of darkness. So we have these brilliant incubation chambers at Cardiff University. And I can put my cryoconite sediments in a bottle with some water to make a kind of closed little cryoconite in the lab and put them under the conditions that I want to, where I control everything in this incubator, playing God a little bit. So on this graph here, you can see the Snowball conditions, which is 12 hours light followed by 12 hours dark to simulate day-night at the equator. And then we have it lined up with the modern conditions where I'm imagining an Antarctic summer which is their growth period, where it's light all the time. And you can see putting them in the dark for 12 hours a day doesn't really seem to bother them. They're kind of following the exact same growth pattern. This is growth measured by the oxygen that they're producing and consuming. So they're following the same pattern as they would be normally. So we can say, having them at the equator, having them in 12 hours light and 12 hours dark, nothing else can change, doesn't make much of a difference to them. So that's another thing that's good to know, means that they might be a good analog to Snowball Earth. Something else I tried was freeze-thaw cycles, which is this graph below. So I thought Snowball Earth, it's going to be harsh. Worst case scenario, they're freezing every night. So I've subjected them to 12 hours of freeze and 12 hours of thaw. Again, this line is a little harder to see, unfortunately, from the way the measurements were taken, but you can see that the two lines, the red line and the blue line, they end up at the exact same place. They haven't died. They haven't gone off growing in some other direction. They're still following the same growth pattern that we see before. So putting them so far in these Snowball Earth-like conditions is not really changing them very much. So that's really good to know that modern Antarctic cryoconites would probably behave not too differently to Snowball Earth cryoconites. Then I thought, can I do even better? Because just as Snowball Earth is not modern Antarctica, Snowball Earth is not in a little bottle. So I've tried to put the cryoconite on some actual ice and get it to form as it would do on the wild. So this is how it looked at the beginning, where I just dumped out some cryoconite into a Tupperware that I brought from my kitchen. to see what would happen, to see if it actually would melt down, because it's not exactly like we can prove how that happens in the wild. No one stays out that long just watching a cryoconite form. And we never know when they're going to form or how quickly. So I think that I'm the first person to do this and actually take measurements. A few people have dabbled, but it's quite time consuming, so I can see why other people haven't, to be honest. Then I bought this much more scientific box from W.H. Smith and set up a proper experiment to measure, to try and get as close to a real cryoconite hole in the lab as I could. And now, as of actually today, when I have finished this set of experiments this morning, you can play a good game of spot the difference. This is one of my open cryoconite holes on the left. This is one I found in Svalbard. This was the first cryoconite hole I ever found. And this is one in the lab. I've now managed to make a nice open cryoconite hole. You can see where it's sunk down, and then it stayed there for a few days. Oops, sorry. Then on the right, you can see an ice-lidded cryoconite hole. This is a picture that my colleague Eva took of an ice-lidded cryoconite hole. And here is an ice-lidded cryoconite hole I made in the lab. After it had sunk, the water in the top was then cooler again because it didn't have the dark sediment there, and it froze over. So I don't know what you guys think, but I think that's pretty good that I've managed to replicate that in lab, and I'm really looking forward to seeing the results a bit more closely. This is what we've got so far from 14 days. Probably just looks like a squiggle because I haven't done any conversion to make it into any useful units. But again, following the same pattern for the first 15 days. So that's promising so far. This is all preliminary results. So that's my question 3. Can we replicate the habitat in a snowball Earth? We seem to have been fairly successful so far, and it seems to be remarkably similar to how they behave regardless in Antarctic conditions. So, question 4, can we imagine a snowball Earth ecosystem? Cryoconite holes don't exist in a vacuum. The material has to come from somewhere and it has to go somewhere. Eventually the glacier will flush out, it will ablate, and they will melt. They will be deposited by streams and by the wind. So they must be connected to other ecosystems. So I'm trying to think about the bigger picture of snowball Earth and what analogues we have for that in the modern world. To look at this, I'd like to talk about my trip last year to Signy Island and the sub-Antarctic. And yes, last year, it was during COVID. I did get stuck and could not get back because everyone's borders were closed. So I did get home 2 months later than planned. So this did kind of dominate my 2020. We can talk about that later if people are interested. Antarctica, of course, is the land of the penguins, the land of the elephant seals and the fur seals, which again, if people want to see pretty pictures, then I have plenty of those. This is Signy Island Research Station. You should be able to see it there. There were 8 people living on the island. There is no cook, there is no doctor, so we had to be very self-sufficient, just the group of us that were there. It's not a very big island, but it is remote and difficult to get to. But the flip side of that is it's very pristine. It is very much like looking at Snowball Earth. It's the closest place that I've ever felt like I have been to looking at Snowball Earth because it's so dynamic and carries so many interconnected ecosystems. You can see this pink patch of snow algae here, which runs into these ponds, gets carried by these streams after it rains. There are everywhere that there is dirty ice, then there is something interesting for me to look at. Where there is water, everywhere there is something brown, there will be microorganisms. And they are in a huge number of habitats, but habitats that are all interlinked and always changing. Even on a daily basis, as the weather changes, they're changing and molding. So what I've been doing for the last month is I have taken different sediments from different habitats. So this is short for cryoconite, glacial stream, ice margin, pond, and stream. So I have these different sediments, and I'm looking at their growth, and I'm going to be looking at their DNA to try and see what the connections are. Where does a cryoconite begin and end? I've put these in the cryoconite situation, I've taken these different sediments, but will they behave the same way as a cryoconite? I want to learn more about what the transitions are like between these different habitats and what the similarities and differences are and what is so special about cryoconite. So yeah, this is the closest I think I've got to seeing what Snowball Earth would have been like on Cygney Island. All of the pictures, by the way, are mine if I haven't said so. That's the end of this bit of the talk, just checking how I did for time. Seems like I think I was pretty okay for 20 minutes. But yeah, like I said, I'm really looking forward to your questions. And I'd like to thank David and the rest of the community for putting my charity up. What it is it's an organization for supporting Indigenous people who are trying to further environmental and social justice. I think that anyone who's been to the Arctic will see that there are Indigenous people who live there and who work there and who are so knowledgeable, and often we've just taken from them and expected them to be there to help us, but they know a lot about the environment, they care a lot about the environment, and so it's really awesome that they have their own organizations to make things better for the organisms and the people there. So yeah, please do support the charity if you can.
Thank you very much, Jaz. That was amazing. Just so everyone knows, if you haven't joined us before, if you do have any questions, feel free to put them in the chat. You should see that along the bottom of your screen, there's a chat button. If you do want to ask Jaz directly, feel free to indicate so, and I can call on you to just ask your question and read it out. Just so you know, we are recording the meeting and we'll publish it on our YouTube page later on. So if you do want to go back and watch anything, then you can do so later on, and just so you're aware. Great. So yeah, I really enjoyed your talk. I've written down a lot of questions, and most of them you've just gone and answered in the next question yourself. So I think that's just a sign of how informative your talk really was. There was something from earlier on that I want to pick up on, just if I may, while everyone else thinks of what they want to ask. So you mentioned that you found this huge diversity of different species, bacteria and eukaryotes and everything else in these cryoconites, and mentioned, I forgot the number, but lots of different phyla as well. I was just curious if you know, how does that compare to other environments like our own microbiome? Just for a benchmark, what's the diversity like?
I will say that the main dominant phyla that we find, like the cyanobacteria, the proteobacteria, they resemble what you will find in a microbial sediment sample anywhere in the world, because those groups aren't extreme loving, they're not extremophiles, they're extreme tolerant, and they're around everywhere. You do have some of the rarer ones that are specifically cold loving, and you only really see in cold environments. As for the diversity specifically, compared to a soil sample, then it's probably not nearly the diversity, but compared to a cold climate sediment, then it's many times more the diversity you would find in other places. But like I say, you can't draw the strict line between where cryoconite begins and ends, because obviously if you have a little stream on the glacier, then you know, maybe it is a cryoconite that melted yesterday. So I'm hoping that I'll be able to answer that question more specifically by my next study, when I'm looking at those sediments from the different areas of Cygney that I showed. When I've done the DNA sequencing on that, then I'll be able to see how they compare to each other, because I don't think many people have done that in the same study, have taken all these different sediments from comparable environments. Because I bet if you took some stream sediment from, you know, the Tamar and you put it in the Arctic or the Antarctic, some stuff would survive, but definitely not all of it. So it's good to look at it in comparison to other cold climate habitats, which is why I showed you those other things like ice algae blooms, which tend to be just dominated by a couple of species.
Cool, thank you very much. In lack of other questions just come through, but I will ask a little follow-up on that way. You mentioned now as well that some of these are more unique to cold water environments, etc. I was just wondering if there's many that are fully unique to your samples that haven't been seen before, any maybe of these that might really excite you as well.
Probably. We have different opinions about this. Sorry, just getting my hair out my face because it's probably annoying everyone, me flicking it around. We have different opinions about this in my team. I have one supervisor who's done quite a lot of sequencing, and he doesn't believe that I'll find anything that he hasn't found before. But there are always things that if you're looking for matches in the database, you only get, the nearest you get is to a family or an order or a phyla. You don't find a match to the species. And it's very difficult to prove whether that is because it's something that's never been found before, something that's never been put on the database that you're using because there's more than one different database, or whether it's something that is just not a very good sequence. It's not a very clean sequence, so it's hard to get a match. So without actually doing a full metagenomics where you're looking at all the genes from all the samples and cataloging them, it's very difficult to say. I think the number of, like, it's very rare that any study covers the number of areas that we have with the same methods. And I'm using high throughput sequencing, which is how I've been able to find way more detail on cryoconite ecology than people have before. So I think it's unlikely that I haven't found some new species, but we'll never know. And one of my supervisors is just always like, I'm sure I found that before. So who knows?
I mean, with the big diversity of microbes, it'll almost be more surprising if you don't find anything new.
Yeah,
right?
yeah, that's what I think.
Yeah. So I'm going to move on to Robin's question, taking this into a bit of a different direction. Robin says you pointed out that the dark specks that launch a cryoconite must have come from somewhere. Do you have any views on potential extraterrestrial origins of falling dust causing us?
There are meteor sites in Antarctica that some people who I work with have studied, and they can be a source of particularly heavy metals. As for organic material, then that is less likely, partly because of the way that things from extraterrestrial objects come into the atmosphere. Then I'm not like I'm not an astrobiologist or an astrophysicist, but a lot of stuff gets destroyed quite easily. So usually all the extraterrestrial things are contributing is some source of minerals and metals around areas where there's a high level of impact. Generally, things are more likely to be from airborne local sources, so within the continent that we're looking at. I'm trying to kind of pump information out of people who look at the transport of microorganisms and the transport of inorganic materials to learn more about it, but it's very much an emerging field, so there's a lot we don't know.
Okay, thank you. Just a quick follow-up. Do you know what the most common origin of these particles might be? If you say they're from the same continent, but I don't know what sort of structures might have existed.
I think it really depends where the cryoconite is, because cryoconite pop up all over the place. A lot of it's near the coast. So I think that it's fair to say, again, this is the problem, that we can't just sit and watch. No one's watched a cryoconite whole form because they just happen around. And it's not like people are sort of like sat out in the middle of the poles waiting for them. And often they're covered by snow and uncovered. So it's very difficult to prove. I think there are people who are trying to trace different parts, particularly of people are tracing the water chemistry, they're tracing the inorganic matter to where it came from, but usually kind of looking at it from the other way, not looking at how much comes from where, but looking at, say specifically, there's someone who looks at airborne material. So they're saying, oh, do we have this airborne material in cryoconite? So a lot of people, just because of the way their research works, are looking at the question the other way. So I feel like I haven't answered the question, but I've told you like the context of how people study it.
Both Peter and Mike have brought us some questions a bit more into the evolutionary angle, which is something else I was really interested in too. I'm going to start with Peter's here. what evolutionary changes can you see over these 100 million years? Are these things just so simple that they don't evolve much? So are the microbes we see now just like the ones back then?
Microbes, in a certain sense, evolve much quicker than animals because they have more ways of doing it. If you take E. coli, it divides every 20 minutes. So the pace that it can evolve at is so much quicker than say, a human being. And also, microorganisms are able to do horizontal transfer as well as vertical parent to offspring transfer. They can just shoot DNA into each other, even if they're not the same species, even if they're not the same phyla, even if it's a fungus and a bacteria, they can pass genes between each other. So microbial evolution happens very differently. I will say that things that live in cold climates work slower. So I don't doubt that a lot of things that were happening before Snowball Earth slowed down during Snowball Earth. And there is a paper that came out a couple of weeks ago that shows some evidence of this in cyanobacteria, that there's a particularly prevalent group of photosynthetic bacteria that seems to show that some of them went extinct and then others became successful. So I wouldn't say that not much has changed in terms of actual changes in DNA. It's just that their function hasn't needed to change, because if you're a microorganism that is in some water at a temperature with certain minerals, then that is your niche. So you don't have a driver to change anything physical, anything practical about your composition. So photosynthetic, say cyanobacteria, probably would have been superficially function-wise very similar to how they were back then. But that doesn't mean they haven't evolved. It doesn't mean they haven't changed. It's just that the changes aren't things that matter functionally. This is the problem with asking a biologist, because what people, evolution isn't just changes that you can detect. Like, a lot of mutations just don't do anything, or they don't do anything noticeable. So yes and no at the same time, and that's not a contradiction.
Thank you very much. In a slightly similar sense, Mike asks, does your work provide any clues as to how complex life forms evolved from this ice ball phase? Also, a quick comment, by the way, very interesting and very effectively presented.
Thank you. I have been thinking about this. Back when I started my PhD, generally you start with a literature review. And so I was getting quite into, theories of like, well, when did the first animals appear? There are some people who seriously think they did appear during Snowball Earth, not after. There's a lot of different ideas about it. So I was getting quite into that in terms of what I read and forming my own opinions. I'm a little behind on that because I've not had the time to read since I've been doing my experiments. So I have my own opinions as a scientist, but I haven't done anything to test it because it's very hard to practically test evolution because it's something that happens over such a long time. I think the people to look for at that are people who are tracing it back from the animal perspective. They're looking, I think things like how I found rotifers and tardigrades, which are animals in cryoconite, is really useful for those people to know. It's useful to know that animals can survive these climates, because it means that they can't rule out Snowball Earth as a habitat for animals. But it is very much that case, because when I started my PhD, there had just been a paper out that said, hey, I'm one of the people who founded the Snowball Earth theory. People are saying to me, that can't have happened, that can't be true, because how would animals have survived? But actually, I think they survived like they do in the poles today in cryoconite. And that was a paper written by a geologist, not a biologist. And that's kind of what my PhD is founded on. So I'm the first biologist to even be giving any proof that organisms could survive on Snowball Earth. So as to looking for how early animals would have fitted into that and how evolution would have fitted into that, that's probably something that future researchers will be looking after I've finished.
Okay, thanks. So early days in that sense.
Yeah, it is very much early days.
Yeah, just out of interest, talking about early animals, how does multicellularity fit into that? Is that something that evolved around the same time as well, or?
Yeah, so I think I want to be quite careful because some scientists get quite upset about definitions of multicellularity, because there were probably biofilms and mats. This is when single-celled organisms work together, but I'm not entirely sure how that relates to multicellularity as we know it in things like animals and fungi and plants, because those groups aren't really related. Like, it's not an evolutionary path. There are bacteria who form biofilms and who form mats. It's not like that's the strict stepping stone to multicellularity in terms of animals. I do think that there is probably merit in looking at what habitats support multicellularity in any form. So I have found biofilms. And basically what this looks like is, instead of just looking like sediment or sediment that's a bit green, it has sort of fluffy bits, it has sort of leaf-like bits, the cells are coming together and building things. And so that is proving that at least in some form of multicellular cooperation, cryoclonite can support that. And it can support animals like tardigrades. But again, how multicellularity developed is a big developmental biology and evolutionary history question that is, yeah, it's related to what I do, but it's so like,
yeah,
it's going to come into the kind of thing I do in a few years when we've got a bit further on these theories.
Okay, great, thanks. I'll move back on to the audience questions then. So Steve asks, do we have any evidence for volcanic activity in the Snowball Earth period? And might then follow out from that have contributed to the formation of cryoclonites?
Yeah, so I do think that, so volcanic ash and similar small particles like that have been found on glacial surface ecosystems, and they can be a form of, they can provide nutrition. Some people think that volcanic activity was a large part of what ended the Snowball Earth, because something had to break this cycle. And if there was volcanic eruptions that were leading to localized warming, more ash in the air, more ash on the surface, you can have a reverse albedo where things are getting darker, so they're getting warmer, and that accelerates and accelerates until you're out of the Snowball Earth. So there is definitely a link there. Again, this is something that people who look at airborne particles are interested in. If you look up Black and Bloom, they are a really cool Arctic research group which one of my supervisors is involved with leading. And they are all about the darkening of the ice sheets in the Arctic. And so that's a mix of pollutants, of bacteria, of volcanic ash, of all kinds of things, and the kind of interrelationship of how darkening glaciers is impacting climate change. And you can apply that to any time in the past, because, you know, volcanoes have always existed.
Awesome, thank you. Just in terms of topics belonging together, I'll skip over to Steve's next question as well, about the atmospheric composition back then, if that was different in any terms of oxygen, nitrogen percentages, and if that's something you've tried modeling in the lab, or that you could try modeling in the lab.
Yeah, that is a really good question. And it depends when in Snowball Earth you're talking about. There was a period of really low oxygen and high CO2, and there was a period of really high oxygen and low CO2. And so you kind of have to pick your time in Snowball Earth to simulate, which I was going to go back and do the reading of and go and do if I wasn't stuck in Antarctica for 2 extra months. I was going to go to the Open University, where they have even cooler chambers than the ones we have at Cardiff, possibly, where you can control the gas very specifically. You can control the pressure. They use them for simulating Mars. And there's someone who's been simulating the Archean, which was a lot longer ago than Snowball Earth. So yeah, the Open University are doing very cool stuff with that. I can semi-control the gas in my incubators. I could also have put them in bags, but now I'm coming to the end of my PhD, and so I will just be writing to people asking if they would like to pay me to do a postdoc on things like that, unfortunately. Maybe someone will give me some money to do it one day, or maybe someone else at the Open University might pick it up.
Always great to have extra options that you can explore with further research.
Yeah.
That's really interesting, actually, that you said there's different periods. There was different periods in the snowball Earth.
Yeah.
So forgive my ignorance on this, but wouldn't this just be already in itself huge evidence that there was big activity changing the atmospheric composition? Or
yeah, so this was mentioned in this recent cyanobacteria paper where they were talking, because I don't know how many of you know this, but more than half of the oxygen in the whole planet is produced by photosynthetic marine organisms, and more than 60-70% by microorganisms altogether. So they're a very big driver of carbon dioxide and oxygen. And so what was happening with the cyanobacteria and algae is very likely to have been driving this. But yeah, I don't want to say too much about it because I'm aware that I haven't caught up in the reading in a few years since my literature review, and I am aware that new papers are coming out all the time. But if you do want to look into Snowball Earth, there are some great documentaries. I really recommend it, and you can follow the kind of path, because it was 100 million years. There were big changes, and there was a deglaciation that probably happened in the middle. So yeah, it depends what phase you're looking at. There were some really high oxygen levels. There were some really high CO2 levels at different points.
That's amazing. You make it sound really thrilling. Going to move on to Robin's question, which I think we've got a bit of a flavor of already, that you work in quite a unique intersection of fields almost. So Robin asks, I'm not sure what to call the particular specialism you work in, but could you give a quick overview of the kind of things some of your colleagues are working on?
Yes, yeah, I'm really glad that you asked that, actually, because I've been asking them for their stories recently, actually, for another talk that I'm doing that contains some fans of theirs in the audience. So my background is in microbiology and molecular biology, and I've always worked on extremophiles, extreme loving organisms. So I started out on salt loving microorganisms and dry loving microorganisms. Then I went to stress proteins in plants, stress response. And then I worked on a species of microorganism that lives in volcanoes at 72 degrees and an acidity of 2.3 pH. And then for my PhD is only when I started working on cold things. But most of my research group at Cardiff is quite interesting. My supervisor, Liz Bagshaw, she is an authority in the field of glacial biogeochemistry. She does a lot of looking at the water and the content of cryoconite in a more kind of chemical side of things. And she has a background in engineering as well. We have an engineer postdoc with us, and they have been in the news a lot for their invention called cryo-egg. Cryo-egg is a round device that you can drop into a glacier, and it will flow through on these fast-moving streams inside the glacier. And while it moves through them, it sends you measurement of the composition of the glacier, of what's going on in there. And that's been on BBC News. If you look up cryo-egg, then you will definitely find it. There's loads of news coverage about it. Since then, they have also developed cryobean and cryoversed. So they're getting the full breakfast items together, because other scientists have contacted them and said, hey, we also like this, but we don't need to drop them hundreds of meters down into high pressure environments. We just want to pop them in a little hole. Can you come up with something like that? So that's when cryobean was invented. And then cryoverse, they said, we have long, thin passages. So can you make a long, thin one? So they did that. And then my newer colleagues, who are PhD students, Elle and Inge, they are working on water, because all the same things in glaciers, the mineral content, the structure, the speed of flowing, are interesting to people who work on rivers, who work on pollution. So they are, particularly Elle is taking hydrobean a water version that doesn't need to be cold tolerant to look at the same things. So I think that my research group in Cardiff is really interesting for just having these great engineering solutions to problems and being able to apply them to a really diverse range of things. Then my group in London are mostly cold climate microbiologists. And like I said, the reason why I'm pulled between Cardiff and the Natural History Museum of London, which I love as a combination of places. They're very different and really cool. I'm in the biology department there, technically in the botany department, because museums are like, oh, algae, that's basically a plant. I get to hang out with biologists there, which is really nice because I'm not a geographer, I'm not a glaciologist, so it's really nice to have a biology department too. Anne has worked on all kinds of amazing microbiology projects. She took some samples from Scott's original trip to the poles. Scott took back some algae mats and Anne ran DNA sequencing on them to see if anything in algae has changed now from when Scott's original trip to the pole was. And she found it's actually pretty much the same, which is, kind of reassuring and interesting. And we have loads of members of our group now who are working on various different things, lakes, lots of microbes in lake stuff. We had one of my colleagues camping on a lake in the middle of Antarctica near the Russian research station. So yeah, I mean, I could do a whole other talk about like all of my research group work, but it is really cool being between a environmental science department and a biology department, because that's where a load of interesting research and collaborations happen. It's also challenging because journals and academia is not always very suited to that. We like our pigeonholes, unfortunately.
Thanks, that's an amazing quick overview. Yeah, as you say, and as I'm sure the audience can understand, it's always some great things happening at the intersection between fields, but also obviously comes with difficulties as well. Robin's actually asking a follow-up question to all that, asking if there's any connection between the biology of stress proteins in plants and the biology of what you're finding now in the cryo world.
Well, yeah, I mean, there is a lot of conservation. We do find the same, I say we, I don't work on this anymore, but there is some tolerance mechanisms are quite old. There's a woman called Patricia Sanchez Bacalado in Bristol, who is trying to trace back some of these stress proteins in cyanobacteria. And although she didn't find any relation between cold tolerance now and cold tolerance in Snowball Earth, because the Earth's been through a lot since then, there have been lots of warm periods, so things were probably lost and then re-evolved. What she did find is that generally response to stress, there are proteins that are just always good. So cyanobacteria that do really well in cold environments are very similar to cyanobacteria in deserts that are surviving dry environments. So because it's all about keeping your nutrients stable, keeping your water stable, protecting your cell. So there are a lot of stress mechanisms that are transferable between different disciplines, different organisms. But yeah, sadly, well, not sadly, I don't regret it, but I have moved away from the molecular side. So I don't kind of see that much of like the deep molecular reasons why my organisms are the way they are.
Thank you. I have two more questions myself, but I'm not quite sure if they actually do fall into your expertise or not, but they are slightly related. I've been wondering around about them for a while, so I thought I might as well fire away and see what you know. So the first thing is about fossils, and you mentioned obviously some single-cellular microbial fossils as well. How does that even work? How can you find them, etc.?
So the most common one that we find are mats. So there are fossilized cyanobacterial mats and algal mats. Usually, the reason that we know that we're cyanobacteria is that there isn't really anything else that forms a mat like cyanobacteria do. The reason that we know that they're algae is because their cells tend to be a bit bigger. You have these generic words like acrotog that are basically just shorthand for this is not a bacteria, this is a eukaryote, but it means they generally don't know any more about that. I think I wound up some people in my department asking everyone, what does this mean to a biologist? Because the people who study fossils are not the same people who study microbiology, and so they're named things that are useful to someone that studies fossils. So they don't really mean anything on a biological taxonomic sense. So we only know very general things about them. This is why most of what we know is from tracing back DNA. So there's such a thing called a DNA molecular clock, where you take two organisms, you look at how many differences they have in their DNA in numbers of base pairs that are different, and you trace it back to try and find their common ancestor based on the average time it takes to make one change. Then you can trace back how long ago those things diverged. So when we do find a fossil that is relevant and we do have an idea that it is, we can calibrate the molecular clock. We can say, okay, well, we know cyanobacteria evolved at least by this point. No, you know, it can't have evolved later than this because this is some evidence of it. So we can use the molecular clock tracing and the fossils together to get a better picture. One or the other on its own doesn't tell you very much.
Okay, interesting, thanks. So basically you can find some general structures that you know are associated with microbes, but nothing more really conserved within that.
Yeah. What we are getting into is biomarkers from lipids. That's kind of the hot new topic. I did a month at MIT in Boston, working on that with their biomarker team. And it's a really cool collaboration because I was just looking at the lipids from my samples to say, hey, I can look at these differences between growth. I can look at these differences between DNA. Is there anything else I can look at to compare and contrast these communities? And people are starting to use lipids as signatures for what communities are like and how they're behaving. So that was a really cool thing to do, especially when those same people who are working on lipid biomarkers are looking at them from fossils to seeing if they can scrub up some lipids that have been preserved and try and like find links. So maybe if I keep doing studies like I'm doing, sending stuff off, and doing it myself to get lipid analysis, and then we have this stuff from the past, maybe one day we'll find two that are the same and we can play snap and start matching those up. That's in the early stages. But yeah, my friends at MIT and Bremen are the people who are into that at the moment, which is cool.
That's amazing. Steve just put up another related question to that, asking how far back in history can we go with ice cores, or do we have any cryoconite history from that?
Yeah, I really need to actually ask someone for some real numbers about this, because this is not the first time I've been asked it, and I'm not an ice core person. I do have in my studies that I've referenced, my ones about the Arctic and Antarctic cryoconite, one of them was from a core. It was a core from Black and Bloom. It is probably in the terms of decades old, rather than the terms of hundreds, thousands, or millions of years old. EGRIP is one of the deepest boreholes that they've managed to make in a glacier that happened recently. And they have been taking cores from those camps in Greenland. When you have a core, then yes, you do find cryoconite, but obviously the further back you go, the less sure you can be that it is cryoconite. There are some kind of signatures, because normally when a cryoconite hole forms, then it forms very clear because you have meltwater forming in sort of quite a neat circle. So you have like the ice is quite kind of white or blue and mixed up, and then you have this sort of clear column. So you can pick up cryoconite, but there's definitely a limit to how far back you can go because ice is so dynamic. You know, no glacier lasts forever. And the climate has changed so much that ice isn't a very good long-term record.
Okay, thank you very much. Questions are slowing down a little bit now. So if anyone does have something else to ask, please do get it into the chat. Otherwise, I will ask my one other question that I'm not even sure if it, as I said, falls into expertise. It is something that I came across a while ago, I think, on one of those Wikipedia sinkholes. So I happened to be reading up on what the oldest or most long-living organism is, and found something about a study that might have been disputed about bacteria living in the ice that are millions of years old, and because they are in the ice so slowly growing, just very slowly dividing or still metabolizing. Does this have anything to do with your research? Do you find any evidence of really slow-lived organisms within your cryoconites or similar environments?
It's definitely way slower than normal organisms, particularly when I was doing kind of like the work that I did for my master's. I was chopping and changing genes and putting them in E. coli to produce genes I wanted to put genes into these extremophiles I was looking at to see what happens. E. coli, as I said, divide every 20 minutes. So I was like, great, grow some, done the next day. Cryoconite can take weeks, months. I've never found the peak of the growth curve in any of my experiments. Unfortunately, it's very difficult to prove if over the winter they are dormant or whether they are active. And I think there probably are cryoconite researchers who have looked at that. Cryoconite is very transient, it melts out on a seasonal basis. It spreads and relocates. It's quite dynamic, as a lot of things on glaciers are. So things deeper in the ice and in lakes would probably be the things that you could have more of a history on, but we definitely see slow growing stuff. And I don't know whether it's, yeah, and again, it's one of those things where these are quite normal organisms that you could find anywhere else in the world. It's just that they're behaving differently and forming part of this particular ecosystem that's so specific.
Okay, thank you. Steve is getting another question in just before the end. So all these micro animals you mentioned, were they part of your experiments as well? I guess there's two different sides to this. Were they something that you found in the sequencing or that you're actually doing further studies on as well?
Yeah, so the micro animals I found all over the place, tardigrades and rotifers. But I have taken them out of most of the figures I've been looking at, because I'm actually now going to have to put them back in, because I have always been working from the angle of Snowball Earth, and we can't yet be sure that any animals had definitely evolved by Snowball Earth. And tardigrades and rotifers most certainly came along later. So haven't really been so interested in them. However, I ended up finding so much out about just the differences in cryoconite between the Arctic and the Antarctic, because no one had used modern DNA sequencing systems to look at that before, that now I'm publishing this paper that's just really about cryoconite ecology, and it's not really anything to do with Snowball Earth. So now I'm putting the tardigrade and rotifer information back in that I had taken out. So I'm going to put that somewhere at the end of the paper in the supplement. But the problem is, if you're looking at DNA, animals are comparatively big, even if they're tiny little rotifers, and they have lots of cells. So a lot of microbiologists kind of conveniently ignore them because you can't compare them to a microbial ecosystem. But they're there and they're part of the ecosystem. They're grazers, really. They're going along eating things, and so they're a important part of it, and so I will be talking about them in my modern paper.
Yeah, cheers. Can I just say some great questions tonight from Steve?
Yeah, thank you for the questions.
Yeah, you're being put through a trial peer review here almost for the paper. Robin did get another one in as well, asking about the body clock, how it seems to be a very early part of the biology for us creatures. But it seems your entities, your microbes, are doing best not to respond to day or night, seeing as you see exactly the same response.
Yeah.
Or even lunar gravitational pull if you're doing it over months. Any thoughts on that?
Yeah, I mean, it's one of those things where in an ideal world, I'd like to keep them running for 6 months. But again, you'd have to pay me a lot more for a lot longer to be able to do that. Yeah, I don't know if because maybe over a longer period of time we would see some difference. But yeah, they seem unbothered. But you have to think about the fact that they are slow growing and they are entombed. They have an isolate over them, and they're used to places that they're on a moving surface. They're on a glacier that moves, and they're in places where you have a whole season of summer. So maybe if you cultivated them, I wonder if you made a culture like they do at culture collections, and you kept it, in the UK or somewhere for years, maybe they develop more of a kind of cycle, kind of a rhythm. But these are organisms that we found from the poles, where you have kind of a timelessness, you know, they go dormant in the winter and then they have a full-time summer. But also I think that, you know, we have to try not to anthropomorphize microbes too much, because I think that animals are very affected by that kind of environmental condition. And obviously there are studies that prove that microbes are too. But we have to try and, you know, we're still at very like early stages of that, I think. There are some cool papers coming out on rhythms of microbes, and yeah, it will be interesting to see what we learn in the future about it.
Awesome, yeah, thank you once again. I think this is a good point to slowly tune out then. Just another comment by Robin, exceptionally fluent presentation.
It's also getting very dark, so I feel like I was like, should I go and turn the light on?
Yeah, so I think at that point, we will thank you very much for your talk. Yeah, I want to agree with Robin. I really enjoyed the presentation. Really vivid. You brought the Snowball Earth back to life for us. So at this point, we usually ask a lot of people to unmute. I think Zoom doesn't let us automatically do that. So I'm going to request to unmute so that we can give a quick round of applause and say thank you to you in person as well. So I will try.
Thank you very much. Thank you for your kind comments. I'm just having a look through them now.